Welcome dear students, we are on the CEC group lectures. We are in the series of characterization of nanomaterials, and today's lecture we discuss the atomic force microscopy, that is AFM. This is the AFM part 1. I am Dr. Sonia Bansal from JC Bose, University of Science and Technology, YMCA, Faridabad. We are dealing with the scanning probe microscopy, where we use the probe to scan the surface. So this is the family of the microscopy forms where a sharp probe is scanned across the surface and some tip and sample interactions are monitored through the display unit. Means in the scanning probe microscopy we use the probe to scan the surface and there is an interaction between the tip and the surface and the sample. So there are the two methods for the scanning probe microscopy, and these methods are scanning tunneling microscopy that we discussed in the previous lectures. In today's lecture we discuss the atomic force microscopy. This atomic force microscopy is the one of the method of scanning probe microscopy, and this AFM, this is atomic force microscopy, means AFM works in three modes. First is the contact mode, second is the non-contact mode, third is the tapping mode, and all are having its own applications. So in today's lecture we discuss these contents. What is the history of atomic force microscopy? Why we use the atomic force microscopy? Means what type of characterization we can do through the AFM. What is the function of atomic force microscopy and the basic principle of the AFM? And what is the basic components used in the atomic force microscopy where we can get the imaging or the surface scanning? So we discuss one by one. First we discuss the atomic force microscopy. Just show you the diagram how it works and what is the part behind it. Now you see on the diagram that here we are using a photo detector that uses that is connected with the feedback control, and this feedback control connected with the piezo scanner, and on this piezo scanner there is a substrate, and this substrate having the sample, means we deposit the sample on the substrate and then we can get its imaging by using the tip. Now you see the tip is on the center and this tip is moved through the laser beam. You are seeing that on the right hand side we are having the laser source, and this laser source fall upon a tip, and you are seeing that the laser light is reflected through this tip and go to the photo detector, and we are having this piezo plate. This piezo plate is moving XYZ direction. Why we are taking it is in XYZ direction? Because it is scanning the X surface, Y surface and Z surface. It means it is moving all the three dimensions. It means we can get the three-dimensional image by using the atomic force microscopy. Now what is the history of AFM? Basically atomic force microscopy is used to investigate the electrically non-conducting materials like proteins. Means we can scan the biomaterials also. So this is also used for the non-conducting materials like protein and other tissues. In 1986 being discovered the AFM by used an ultra small probe tip and at the end of the cantilever. This is the cantilever which plays a very important role in the AFM, and this cantilever contains a tip, and this tip in 1986 being explained a ultraviolet small probe tip which is connected with the cantilever. But after that in 1987 we can explain this cantilever in the form of the vibrating form. So he published the paper in 1987 in the Applied Physics, and he discovered that AFM setup with a vibrating cantilever technique, which use the light lever mechanism, means this cantilever tip can move or vibrate through the surface of the specimen, and it can get the good imaging and good picture of the surface. Now, why we use the AFM? What is the function of AFM? AFM is a versatile tool to investigate topography of the surface. As we are saying that we are scanning the surface, when we can get the topography or topological structure of the surface. We can find out the properties of the surface. Properties of a single molecules also can be get through the AFM. Forces with the molecules are attached with the forces that can also be find out by using the AFM. But always consider experimental conditions and artifacts on the measurement. Means this measurement can be done by using the different experimental conditions or at the different parameters. Means we can get the scanning of a surface in the air as well as in the water as well as in the oil. It means we can change the medium also, and we can get the imaging of the surface. It means experiment conditions can be changed, and we can scan the surface and we can find out the properties of the surface. We can get the topography of the surface. So these are the basic applications of the AFM, and AFM are used to investigate these properties of the surface and the forces within the molecules. Moreover, we can scan the single molecule by using the AFM. Next, what is the function of atomic force microscopy? As we discussed in the previous lectures, like if we talk about the scanning tunneling microscope, we used the quantum mechanical effect of tunneling because we discussed about the quantum mechanical effects. But here in the atomic force microscopy, we use the atomic force which map the tip and sample interaction. It means by using the atomic forces, we can scan the surface by interacting the tip and the sample. The force is changing, and the tip which is attached with the cantilever is can vibrate. So this atomic force microscopy give the mapping or give the imaging of the sample by interaction with the probe or by tip. Now, often referred to as a scanning probe microscopy, SPM, there are the atomic force microscopic techniques for almost any measurable force interaction. As we discussed that it is the atomic force microscopy used to find out the forces between the molecules. So this is used to find out or measure the force interaction like van der Waals, electrical and magnetical thermal properties we can find through this AFM. Next, instead of using the quantum mechanical effect of the tunneling, forces are used as a map, the tip sample interaction. Similarly, we can find out the forces. Then what is the basic principle behind the AFM? AFM is an analytical instrument used to study the surface structure of the solid, which include the insulator, non-conducting material, in which we can find out the surface of protein and other bio tissues. So this AFM used to get the surface imaging, and the surface texture and the properties are obtained by detecting the weak atomic interaction forces between the specimen surface and the microforce sensor. What is this microforce sensor? This microforce sensor basically a tip which is attached with the cantilever. So we are discussing that this cantilever is attached with the tip, and this tip scan the surface, and this work as a micro force sensor. Basically we are calculating the force between the tip and the sample. How we can get the, how can we calculate the force? It means it should in the contact, or it should not in the contact, but it must be very near to the surface, or it must vibrate on the surface. That can be, this tip is attached with the cantilever, and the cantilever will decide the deflection of the tip, and this tip scan the surface. So this is the basic principle of the AFM, is that it find out the surface texture as well as the forces between the specimen and the probe. That is also known as the micro force sensor. Next, working of AFM. What is the work? How AFM works? The principle of AFM is that this nanoscale tip is attached to a small cantilever. Now here we see that this tip is of the size of the nanoscale. That's why it can scan the single molecule. So we use the nanoscale tip which is attached with a small cantilever which forms a spring. Why we are talking about it work as a spring? Because this will vibrate at the surface of the specimen, or it will move through the surface. So this cantilever is used to make a tip as a spring, and as the tip contacts the surface, the cantilever bends, and the bending is detecting using a laser diode. Means we are using a laser beam to bend the cantilever. We fall the laser beam on the cantilever. This cantilever gets the force and it bends on the surface. And split photo detector. The bending is indicative of the tip sample interaction forces. It means when a laser beam fall upon a cantilever, this because this tip is attached with the cantilever, this laser beam exerted a force on the cantilever tip, and this force is interact with the sample, and so we are having the tip sample interaction force. This bending is indicative of the tip sample interaction force by using the laser beam. Now you see in the diagram, you see on the right hand side there is a diagram where we are using the laser beam which is fall upon a cantilever, that is notation by the probe. The laser beam falls upon a probe and it exerted a force on the cantilever. That's why this cantilever move downwards and this tip move on the surface of the sample, which is on the piezo element, this which can move up and down the surface of the specimen, or the specimen which is on the piezo element can move up and down, and this tip is up and down by using the laser beam. Now you are seeing in the diagram that this laser beam is reflected towards the photodiode, and this photodiode gives a result of all the forces changes and will present the imaging of the sample. A pair of cantilever with extremely sensitive weak force is fixed at one end, and at the another end a tiny tip is brought close to the sample, then interacts within. Means we are exerting a very weak force on the probe, and because of this weak force, probe move towards the sample, and there will be the interaction between the sample and the tip. Tip is as a microsensor, so it will scan the surface, and here we see that the reflection can get in the in the photodiode. Next is the the force will make the cantilever be deformed or motion status change. As the laser beam fall, force will make the cantilever be deformed or motion status changes. What is the effect of the force on the surface that we can be recorded. This can be recorded. After occurring the interaction between the tip and the sample, the cantilever will swing. As the force exerted on the cantilever, it will scan the surface and it will move towards the surface, either in the contact mode, either in the non-contact mode, or in the tapping mode. So after interaction between the tip and the sample, this cantilever will swing, and that will end under the rest. So when the laser irradiates at the end of the cantilever, the position of the reflection light will change with the swing, and this will resulted in the generation of the offset, and we can get the result on the photodiode, and the scanning of the surface can be recorded on the photodiode as the force exerted on the cantilever. And the change of the position of the cantilever because of the reflection of the light can be find out on the photodiode. Then offset is recorded and converted into the electrical signal by laser spot position detector, as it is recording the force points. So after that it will convert it into the electric signal by the laser spot position detector, and by using this we can get the imaging of the surface or the scanned area of the surface. Now when the scanning the sample, sensor can detect these changes, how the laser beam force the probe, and this probe scan the surface, and how the force between the probe and then the sample changes, that is recorded. When this scanning takes place, the sensor can detect these changes by obtaining the force distribution information between the surface and the tip, and we can get the surface morphology, surface roughness information by using the movement of the tip of the cantilever. So by scanning the surface, by putting the laser beam on the probe, and this scan the surface, and that can be recorded, the value of the forces and the changes in the sensor can be detected by using the photodiode, and by using this we can find out the surface morphology, surface topography, surface roughness information, defects in the surface, defect in the devices that we can measure by using this AFM. Now basic components of atomic force microscopy are cantilever and the tip. The cantilever containing the tip. Second most important is the laser that exerted a force on the cantilever. This cantilever move on the surface. This tip is attached with the cantilever. This is like a micro sensor, and this is of the nano size. Scanner that control the XYZ position of the tip. Detector that detect the output. Feedback control unit. This is used to control the movement of the vibration of the cantilever. Display unit which give the imaging of the surface. On the right hand side, if we collect all these, then we see that here we are seeing that sample is on the sample holder. Laser beam fall upon a cantilever tip, and this cantilever tip comes closer to the sample, and there is a photodiode which, or we can say it is a detector, which can detect the force changes or the changes in the tip or the cantilever that can be recorded. And laser is the most important, which give the force to the cantilever to go closer to the sample. So these are the basic components of the atomic force microscopy. We discuss one by one. What is a probe? Probe, it is a nanoscale tip, and it is made of silicon and Si3N4, and only part that contact sample. Shape should be critical. Resolution depends on it. The shape of the tip decide the resolution of the surface. Means we can get the good resolution if we are having the good probe. So probe is very important to get the good resolution of the surface. Here in the diagram we are seeing that there is a tip which is attached with the cantilever, that is a probe. Second is the cantilever. Cantilever is designed with a very low spring so that it is very sensitive to any force, because we are giving a very minimum force to the cantilever, so it should be so sensitive that so that it can move or vibrate by using the by using this force. The spring system, force sensors are used. Bends is presence of the attractive repulsive forces as a laser beam fall upon it. Cantilever deflection converted to the forces, and we use a Hooke's law in defining the cantilever deflection, which convert into the forces. Next is piezoelectric transducers. This is like a scanner which move in the XYZ direction and is controlled by the piezoelectric tube scanner. Next is this is made of the lead, barium, titanate and zircon titanate. Though these piezoelectric transducers are used for the movement of the sample which is on the substrate. And feedback control, this is used to maintain the fixed force between the probe and the sample. This will move the probe on the surface, and this is the combination of all the instrument which are used, all the parts which are used in the atomic force microscopy. Thank you.